Hey guys, my name's Arujin, and today I'm going to show you guys how to simulate a model of a protein. So the protein that I chose is Listeria lysine O, as it has a big impact in nanomedicine. So Listeria lysine O is a protein that creates pores in the cell membrane, eventually leading to cell death, and this can take only around 10 to 20 minutes. So the first thing we're going to do is copy the name of the protein, and then go to a website named UniProt. So in UniProt, we're going to paste the name, we're going to click enter, and we're going to get a couple of results. We're going to make sure to click the right one, which is in this case the first one. And as you can see here on the left hand side, there's a lot of things like function, names, and taxonomy, but we're going to skip directly to sequence. And as you can see here, sequence is just a listing of the amino acid code, which in this case is 529 letters. So the more complex a protein is, the greater the amino acid code length is. So as you can see here, Listeria lysine O is a pretty complex protein. So what we're going to do with this amino acid code is we're going to copy the sequence, and then we're going to go to something called AlphaFold, which is a AI model to predict the structure of many biological molecules, or in this case, proteins. So we're going to paste the sequence right here, and as you can see, pretty long. And then we're going to click confirm and submit job. So it's going to take around 2 minutes for AlphaFold to take all this information and decode it. So we're just going to skip past that step as I've already done it. And as you can see here, this is what AlphaFold thinks the protein is actually going to look like. And there's three important things to note here. So the first thing is the color, which denotes how confident AlphaFold is that this is actually what the protein looks like. So the dark blue means that AlphaFold is 90% or greater confident. And then as you can see here, that's majority of the protein. So AlphaFold is really confident that this is what it looks like. Then there's the cyan part, which is 70 to 90%, which is just a tad bit around the protein. Then there's the yellow part, which is 50 to 70% confidence, which is just a tad bit here. And then finally, there's this orange stripe, which is less than 50% confidence. So in total, we can see that AlphaFold is really confident with this protein as it's majority dark blue. And in total, it's an 86% confidence rate, which is really high. The second thing that's important here is aligned residue versus scored residue. So aligned and scored residue are just a measure of how similar this protein, Listeria lysine O, is similar to another protein. So aligned residue is how similar the genetic code or amino acid code or how the protein functions to another protein. So the higher the aligned residue, the greater similarity this protein has to another protein. And then there's scored residue, which is basically if you stack two proteins on top of each other, how similar would they look? And it's the same thing as aligned residue, the greater, the more similarity. So that's this chart here. Finally, there's expected position error, which is measured in angstroms. So angstroms is a really small unit, which is about 1/10 of a nanometer, so extremely small. So we can see these numbers from expected position error from around 0 to 22 or 23 angstroms, which you can see is really, really small. So we can see how accurate AlphaFold actually is with this protein and how confident it is. So what can we actually do with this protein? So with the model, we can actually predict how this protein will interact with other proteins and how that will affect its function or its job. And we can use this for multiple applications. And for example, in nanomedicine, we can implement that information into a nanorobot and hopefully try and replicate the job of a protein. So thank you guys so much for listening. And that's how you decode and make a model of a protein. Thank you.